Good morning. This is the first video on the cell. And on this slide, you can obviously see that we have three types of cells. And the first two types of cells to the left are eukaryotic cells. And we are going to review together what a eukaryotic cell means and what the characteristics of a eukaryotic cell are. So here you can see a plant cell. There you can see a diagram of an animal cell. So cell number 1 is a plant cell. Cell number 2 is an animal cell. And cell number 3 is a cyanobacterium. So it is a bacterium or a bacterial cell. Of course, as I said at the beginning, the first two cells, number 1 and number 2, are eukaryotic cells. They are eukaryotic cells. Whereas the third cell is a prokaryotic cell. What is a eukaryotic cell and what is a prokaryotic cell? We are going to discuss this in detail on the following slides. But as a start, we can split the word eukaryotic into eu, which means true, and karyotic, or karyo, which means nucleus, which refers to the nucleus. Here we have the same word karyo, which means nucleus, but at the beginning we have pro, and pro means before or first or primitive, as you like. So it's obvious from the nomenclature that scientists consider prokaryotic cells as cells coming earlier or having existed earlier on Earth before the appearance of eukaryotic cells, and that there is a difference between eukaryotic cells and prokaryotic cells, because the term true here refers to the nucleus. So it is important. The major characteristic is that eukaryotic cells have a true nucleus, whereas prokaryotic cells or bacteria, they do have genetic material in the form of DNA, but they do not possess a true nucleus, and this is what we're going to explain in detail. Obviously, because the prokaryotes came first and because they just have DNA but not organized in the form of a well-developed nucleus, so it's obvious then that according to scientific hypotheses, that the bacteria and the prokaryotes in general came before the eukaryotes, or at least are less developed than the eukaryotes. And here we have the theory of endosymbiosis. The theory of endosymbiosis is important as a concept, and you are frequently asked about the theory of endosymbiosis, whether in MCQ or in free response question. This theory assumes, as we have said before, that all organisms at the beginning of life or early on Earth were prokaryotes, and that's why we call them here in the diagram ancestral prokaryotes. And what you see here is a cross section in the prokaryotic cell. So scientists assume that this is just a hypothesis, this is just a theory, but it's not, of course, definitely proven. There are criteria, or there are certain pieces of evidence that help strengthen the theory of endosymbiosis, and we are frequently asked about these pieces of evidence. Let's see how scientists visualize the theory of endosymbiosis and how they use this theory of endosymbiosis to explain how we can start starting of a prokaryote, we can reach a eukaryote at the end of the day. So what happens first is that there is infolding or invagination of the cell membrane inward. So this is what happened here, what happened there. And by having this done, you will have double membranes here. And those double membranes they resulted from infolding. This kind of folding, we call it infolding because it is folding towards the inside of the cell. And this infolding resulted in segments of double membranes. And of course, these segments of double membranes are very similar to the plasma membrane, meaning that they are phospholipid in nature. And we ended up, according to this theory, by having a true nucleus that is surrounded by a nuclear membrane. So the most important characteristic of a true nucleus is that you have the genetic material inside, and that genetic material is surrounded by a double membrane. This is the first change that occurred in the ancestral prokaryote. And then here we have two prokaryotic organisms, or two structures that are considered prokaryotic organisms that try to invade the original prokaryotic cell that now has a true nucleus. And these two organisms are essentially prokaryotic organisms. So actually, in this third diagram, we just have a prokaryotic organism that is trying to invade another prokaryotic organism. This is the original prokaryote. This is the original one with which we have started. And then this is a bacterium that is called cyanobacterium. And that cyanobacterium is blue-green. And it is blue-green because it contains pigments, and these pigments can absorb light. And therefore, this organism can carry out photosynthesis. It can carry out photosynthesis. And the other prokaryotic organism is another bacterium. That other bacterium does not have the pigments that are present in cyanobacterium, but it is characterized by being aerobic, meaning that it can carry out aerobic cellular respiration. What is aerobic cellular respiration? That you can, or this organism can break down a glucose molecule in the presence of oxygen, to get energy. And this energy, or the release of energy, will be associated with the release of two side items, which are carbon dioxide and water. The main purpose of that chemical reaction, which we call cellular respiration, is the production of energy. It's not the production of carbon dioxide or water, but it is the production of energy. So this is our main purpose, and this energy, of course, will be utilized for all vital active processes in the body. An active process is a process or chemical reaction, an action in the body that requires energy and cannot do without energy, like muscular contraction, for example. And it is a vital process because it is, why do we call it a vital process? We call it a vital process because it is essential for life. And it is postulated or hypothesized that these prokaryotes intruded themselves inside the original prokaryotic cell. And after all these changes, the scientists assume that the original ancestral prokaryotic cell is no more prokaryotic, but it has been converted into a more complicated and sophisticated cell, which is a eukaryotic cell. Eukaryotic because it contains a true nucleus, but you can see from the diagram that it's not only about the true nucleus, but it is about those minute structures that were intruded in the cell. And scientists assume that these cyanobacteria will allow this original cell that hadn't any pigments before to photosynthesize, and that these are the green plastids or the chloroplasts inside the plant cell, whereas these aerobic bacteria that can carry out cellular respiration efficiently, and I forgot to tell you that aerobic cellular respiration means that this process of cellular respiration that I have just explained should occur in the presence of oxygen. And now these tiny organisms are considered mitochondria. So we have started by a prokaryotic cell that was rather simple, having no minute structures inside, into this eukaryotic cell that is now containing a nucleus and containing these structures. These structures, whether the mitochondria, what we consider now mitochondria, or what we consider plastids, are characterized by the following. Number one, they are small structures. Number two, they are membrane-bound structure. This is very important. You will be using this frequently. They are small structures. They are membrane-bound structure. Number three, they help specialization through compartmentalization, because every small membrane-bound structure is surrounded by a membrane and represents a small different compartment. So this is what we call compartmentalization. Compartmentalization. Okay, compartmentalization. And this compartmentalization actually helps specialization, because each of these minute structures, or each of these compartments, will have a specific function. For example, the green ones having the green pigment will be specialized in photosynthesis. The ones that we see here, orange-brown, will be specialized in energy production. And when you have specialization, the more specialized the cell is, the more efficient it is. And therefore, it's just like when you go inside a hospital, for example, if all the medical doctors are spread over the hospital without specifying where, for example, the surgery department is, where the dermatology department is, where the internal medicine department is, therefore you will waste a lot of time looking for the doctors you want. But if we divide the hospital into compartments, into departments, and every department into rooms, and every room has a label on it with the name and the specialization of the doctor or the physician, then it saves time and helps more efficiency of that medical institute or of that hospital. Okay, these structures that increase the efficiency are called organelles. So all the characteristics I've mentioned before are characteristics of organelles. They are small, they are membrane bound, they help compartmentalization, and therefore specialization and help efficiency of the cell. And this explains to you why eukaryotic cells are more efficient than prokaryotic cells. So we can here highlight this question or highlight this concept that eukaryotic cells are more efficient than prokaryotic cells. Of course, if my body, like in human beings, if the body of the living organism is made up of eukaryotic cells, then it is a eukaryotic organism. You can say a eukaryotic organism or a eukaryote. So a eukaryote is an organism that is made of eukaryotic cells, and similarly, a prokaryote is a prokaryotic organism. The appearance of such more efficient eukaryotic cells led to the appearance of higher, more developed organisms, which are the eukaryotic organisms, and allowed the living organisms to move from the level of single cellular organisms, because all prokaryotes are one-celled or unicellular. We used to say before unicellular, and now we say one-celled. So one-celled organisms. And by the way, all bacteria are prokaryotes, and all prokaryotes are almost bacteria. So they are synonym to each other, almost synonym to each other, okay? So the appearance of eukaryotic cells has allowed the appearance of eukaryotic organisms, and eukaryotic organisms are multi-celled organisms or multicellular organisms. But the multi-celled eukaryotic organisms are themselves divided into lower and higher. The lower have a smaller number of cells and can carry out a smaller or simpler functions, whereas the higher organisms are more complex and can carry out more complex functions. You are sometimes asked about the evidence that supports the theory of endosymbiosis. So the most important evidence is that, of course, the cyanobacterium at first is a prokaryote. So it has one membrane surrounded it, one membrane surrounded it. But after having pushed inwards, it has pushed with it, it has taken with it part of the cell membrane of the host. So here we will end up by having a double membrane. So the most important evidence is that these organelles that are the chloroplasts, or specifically the green plastids, the chloroplasts, and the mitochondria are double membraned. You will know very soon that these organelles, these small structures, are double membrane. So they have their original membrane that was present from the beginning, in addition to that that second membrane that has been acquired upon invaginating the original host cell, upon invaginating the original host cell. So this is the first evidence that supports the theory of endosymbiosis. The second evidence that supports the theory of the symbiosis is that both chloroplasts and mitochondria, and chloroplasts and mitochondria in particular, are they do have their own DNA. They have their own DNA that differs from the main DNA of the eukaryotic cell, meaning that if you look at that eukaryotic cell, you will find that this is the original DNA, but meanwhile, this mitochondrion has its own DNA, and that plastid has its own DNA, and that mitochondrion has its own DNA. And that DNA is in the form of small, circular, multiple DNA molecules. So it is characterized by being, the DNA molecules are small, they are circular, and they are many, they are multiple, okay? And not only this, but of course, what's the importance of them being of the DNA in the chloroplast and in the mitochondria being circular? It is very similar to the single circular DNA molecule of the original prokaryotic cell, because here it is a single DNA molecule that is circular like this, prokaryotic, and in addition to this, the prokaryotic cell has multiple DNA molecules in addition to the main or central DNA molecule that are called the plasmids. So scientists found out that inside the green plastids and inside the mitochondria here in eukaryotic cells, there were plenty of small circular multiple DNA that are very similar to plasmids. Not only this, but these plasmids can be replicated. These plasmids can be replicated on their own, without waiting for the main DNA of the cell to be replicated, without waiting for the whole cell to undergo cell division, without waiting for the whole cell to undergo cell division. Okay? So, but there are, of course, things that are against. For example, how come that if this is, as scientists assume, this original cell was a prokaryote, how come that the single DNA molecule became multiple DNA molecules? Because we know that in eukaryotes, we have multiple chromosomes, and chromosomes are DNA molecules, and whose number differ from one organism to another. So you are frequently asked about this kind of evidence. And now we are going to talk about the modern cell theory. Of course, the evidence is not written here. It's written elsewhere. But please note that you have you have to know this or those pieces of evidence that support the theory of endosymbiosis that we have just explained. This is very important. And now we come to the modern cell theory. The modern cell theory has been postulated by Schleiden and Schwann in 1838. And this modern cell theory states that all living organisms are composed of cells, or at least this postulate of the modern cell theory has been postulated by Schleiden and Schwann in 1838. We will not ask you about who postulated the theory. So the cells are the basic units of all living organisms. And up till now, we can say that they are the basic structural units, the basic structural units of the living organism, because living organisms are composed of cells. We will see later on, of course, that cells are not only basic structural units, but they are also functional units of the living organisms. In 1855, 17 years later, Virchow stated that all cells in the living organism arise from pre-existing cells. And this supports the theory of creation of living cells. But when Virchow says that they come from pre-existing cells, he meant that they arise from cell division. They arise from cell division. So if you have a living cell, then this originally appeared as the result of mitotic, for example, division of an original cell. The original cell, the original parent cell, is usually called a mother cell. So, and the two newly formed cells are called daughter cells, independently of whether this cell comes from the mother or the father. Okay? And most animal cells and plant cells have a diameter that ranges between 10 microns and 100 microns. Of course, you all know that a micron is 10 to the power minus 6 of a meter. So a micron is a micrometer, and a micrometer is 10 to the power minus 6 of a meter. For example, human red blood corpuscles are only 8 microns, so they are very close to 10 microns, but they are one of the smallest cells in the body. Although red blood corpuscles are not, when they are mature red blood corpuscles, they are not considered true cells. And that's why we say corpuscle. We don't use the term cell. Corpuscle means corpuscular or spherical, but we use the term corpuscle to avoid saying red blood cell. Why? Because you will see during your study of the cell that red blood corpuscles do not have nuclei, just to give more space for accommodation of more of the red pigment hemoglobin that is important to carry oxygen to the cells. As we said before, cells are divided into two major categories, the prokaryotic cells and the eukaryotic cells. And we also said on the previous slide that all bacteria are prokaryotes. We also said that eukaryotic are more complex and that most forms of life are formed of eukaryotic organisms or eukaryotes, and that human beings, of course, are eukaryotes. And now we come to that table that represents the major differences and similarities between prokaryotes and eukaryotes. And when we say prokaryotes and eukaryotes, we mean organisms. So prokaryotes are organisms that are made of prokaryotic cells, and eukaryotes are organisms that are made of eukaryotic cells. We have just said that in eukaryotes, this is of course one of the major determinants, is the presence of internal compartments or compartmentalization, and this is through the formation of organelles, as we said. So if prokaryotes contain no internal compartments, yet eukaryotes have distinct membrane-bound organelles. And as we have also said on the previous slides, we said many pieces of information on the first two slides, that those membrane-bound organelles may be surrounded by a single membrane or a double membrane. Most organelles are surrounded by a single membrane, with few organelles only that are surrounded by double membranes like the mitochondria and the chloroplasts. The mitochondria and the chloroplasts. The nucleus is a double membrane structure, but there is controversy whether the nucleus can be considered an organelle or not. In my opinion, the nucleus should be considered an organelle because it makes an internal compartment of the cell, and it plays a major role in the cell, and it has very specific important functions in the cell. But whenever you're, for example, asked to list 3 organelles on your test and explain their structure and function, try to talk about 3 organelles other than the nucleus, or write 3 organelles, explain 3 organelles, and then add the nucleus, because you can never know what the reader thinks about thinks of the nucleus. Number 2, when we talk about the differences, so in prokaryotes, we have no internal compartments and we have no nuclear membrane. But here, of course, we have a nuclear membrane. Number 3, there are no organelles. So you have no endoplasmic reticulum, no mitochondria, no vacuoles, no chloroplasts, no lysosomes, no centrioles, no Golgi bodies. What are these? These are specialized organelles. These are specific, or I prefer specialized organelles. And this is a good habit, that every time you say organelle, you say specialized organelles to make the readers know that you know that organelles increase specialization and efficiency in the living cell. And of course, since they don't have a nuclear membrane, so their nuclear membrane is naked. So I would rather say that this is number one and this is number one, because all this is related. In eukaryotes, we have distinct membranes, and as we said before, this makes them more specialized to perform specific functions, and I would rather use a more scientific adjective, which is specific function. And what's the importance of this? To increase cell productivity. How does specificity increase productivity? By increasing the efficiency of the cell. The only organelle that is present in prokaryotes is the ribosome. So be careful when you say that prokaryotic cells do not contain any organelles, to mention that the only exception is the ribosomes. So ribosomes are present both in prokaryotic cells and in eukaryotic cells, but they are smaller in prokaryotic cells. First of all, why are ribosomes the only organelles present in both? This is because ribosomes carry out a very important function that all living organisms, whether they are prokaryotic or eukaryotic, require, and this is protein synthesis. So ribosomes are the site for protein synthesis. Why is that function so important? Because proteins are essential for the cell. Proteins are essential for formation of enzymes. They are essential for formation of hormones. They are essential for cell division, and so on. Sorry. Now, the third difference between prokaryotes and eukaryotes is the type of chemical reaction, specifically cellular respiration. So metabolism, what is metabolism? Metabolism represents all the biochemical reactions that occur in the cell. But here, when we say metabolism, we are more talking about or focusing on cellular respiration, because it is cellular respiration that is mostly divided amongst all the metabolic reactions in the body. It is cellular respiration that is divided or classified into aerobic, occurring only in the presence of oxygen, or anaerobic, occurring only in or occurring in the absence of oxygen. Don't say only, please, because there is something I'll be explaining now. And it is important here to differentiate between when we have an aerobic organism, when we have an aerobic organism. There are obligatory aerobic organisms, or more simply, obligate aerobic organisms, meaning that they can carry out cellular respiration and produce energy only in the presence of oxygen. And there are facultative aerobic organisms, meaning that they are essentially anaerobic and can work in the presence of oxygen. Mostly when we say facultative aerobic organisms, this originally means that they can do both. But facultative aerobic organisms, they are organisms that can respire both aerobically and anaerobically. Similarly, When we have an anaerobe, it might be an obligate anaerobe, meaning that it only respires in the absence of oxygen, or it might be a facultative anaerobe. So it's essentially the same. Facultative means that under certain conditions, the organism can respire in a different manner. And it is usually an aerobic organism that can convert its metabolism into an anaerobic organism. For example, here, when we look at the eukaryotes, the metabolism is mostly aerobic. So, but some eukaryotic organisms like yeast, for example, and yeast, by the way, is a one-celled fungus, they can respire anaerobically through alcoholic fermentation. But even inside the human body, muscle tissue can respire anaerobically under certain conditions in severe muscular exertion when the input of oxygen to the muscles is less than what the needs of. So although a facultative aerobe and a facultative anaerobe seem like they represent the same thing, but when we say facultative, I'd like to highlight this. When we say a facultative aerobe, this differs, or there is a slight difference between a facultative aerobe and a facultative anaerobe. What does this mean? A facultative aerobe means that it will be sometimes anaerobe. And this indicates that it is essentially an anaerobe. So a facultative aerobe means that this organism can choose to be aerobe, to be an aerobe. So essentially, it was an anaerobe. So a facultative aerobe is essentially an anaerobe that respires in the absence of oxygen, but can, under certain conditions, become anaerobe. A facultative anaerobe, this means that this organism, the facultative anaerobe, can choose to be an anaerobe. So it is essentially an aerobe, but it can be converted into an anaerobe under certain conditions, for example, when oxygen is not available. And then, so this is again the first difference. So this is again the first difference, second difference, third difference, and then fourth difference. But before discussing the third difference, I'd like to say that when yeast, which is a one-celled fungus, decides to respire anaerobically, this is called alcoholic fermentation, because there is ethyl alcohol that is formed, alcoholic fermentation. And this is, by the way, what yeast do when they are in dough, for example. When we make dough for pizza or for whatever bakery, the yeast respire anaerobically inside the dough, and they produce ethyl alcohol and carbon dioxide and energy. So they produce carbon dioxide, they produce CH3, CH2OH, which is ethyl alcohol, and they produce a small amount of energy that is enough for them, which is 2 ATP molecules per glucose molecule. And that explains why we put sugar with yeast. Because yeast, when your mom, for example, prepares dough, she adds sugar to the yeast, because the sugar we use on daily basis is sucrose. So the yeast will break down that sucrose into glucose and into fructose, and then will use glucose as a source of energy. Every molecule of glucose, every molecule of glucose will produce a small amount of energy, which is 2 ATP. So you have to know that anaerobic respiration is usually done by primitive organisms, even if they are eukaryotic, but mostly one-celled, because it produces a small amount of energy. So this is the first thing to know. We now understood why we add sugar to yeast. The second thing is ethyl alcohol. And this explains why when you put yeast on dough and leave dough for a long period of time in a warm atmosphere, your dough might acquire the smell of alcohol. And it is an unpleasant smell to be found in dough, of course. So you shouldn't leave it more than the time recommended. The third thing is that it produces carbon dioxide, and this is important to elevate your dough in when it is put in your oven. On the other hand, in eukaryotic organisms, we never see alcoholic fermentation. Fermentation is almost anaerobic respiration. They are not synonymous, but fermentation is anaerobic respiration plus something, and we will see this later on. Now, anaerobic respiration is almost fermentation. Anaerobic respiration is glycolysis, that will be explained very soon, plus an additional step. We will be explaining this very soon, so don't bother yourself trying to understand it now. On the other hand, if we have eukaryotic organisms, they never do, or eukaryotic tissues, they never do alcoholic fermentation, but they carry out acidic fermentation. So when a muscle does not receive enough oxygen and respires anaerobically, it will produce lactic acid, and it will produce carbon dioxide together with 2 ATPs. And that's why, first of all, anaerobic respiration in the human body is painful because lactic acid stimulates pain nerve endings. Secondly, the 2 ATPs are not enough, and therefore we consume a lot of glucose to get the required energy. And that's why when you reach the state of anaerobic respiration, the status of anaerobic respiration, this helps you lose weight. Number 3, when you rest, this lactic acid will be washed away by the circulation, and therefore the muscle cramps and the pain that were resulting from lactic acid will disappear. The fourth difference is the cytoskeleton. What is the cytoskeleton? We will discover very soon that inside the eukaryotic cells, you have certain filaments, which means elongated protein molecules. And these elongated protein molecules, or even the smaller microtubules that look like very thin chalumeau or very thin straws like this, these are protein molecules, and they are present inside the cell to give support to the cell, to give structural support of the cell to the cell. But we will understand very soon that these cytoskeletal proteins are not there only to provide structural support to the cell, but they can also provide, they can also give function to the cell. In prokaryotes, the cytoskeleton is absent, but if there are microtubules, so these microtubules will only be there to power cilia and flagella. For example, certain prokaryotic organisms like bacteria have a flagellum that helps them. Most bacteria, of course, are non-flagellated, but this flagellum will help the bacterium to move, for example, in tissue fluids or whatever liquid environment they are present is. So if you look at a section of that flagellum under the microscope, you will find that you have microtubules. For example, you can have 9 pairs of microtubules like this. This is a section, by the way, etc. And in the center, you might have a central pair. So this is the structure of the flagellum under the microscope and the structure of the cilia. What are cilia? There are prokaryotic organisms that are ciliated, and cilia are essentially made of microtubules, but these microtubules are short and many, and they help movement in the flagellum. Or in the case of the flagellum, we have only one that is long and singular. Actually, in order to make things more accurate, because this table is actually a picture or a photo, an image that I took from Google Images, but here it's written that the cytoskeleton is present in the form of actin and myosin filaments. This is applicable in the case of muscles. For example, we have actin and myosin filaments that are arranged in a special regular way in the muscle, and that are contractile, and they help contraction and shortening of the muscle. But we also find actin in other cells to support the cell membrane. So under the cell membrane, you will find actin filaments like this. So it's not only in muscles that you can find actin filaments, but myosin filaments are mainly present in the muscles, although they might be present in other muscles, and we will see this in detail. Bacteria are flagellated, but most ciliated organisms are unicellular eukaryotic organisms. For example, in case of the paramecium. The paramecium has a true nucleus. It actually has 2 nuclei, a macronucleus and a micronucleus, and it shows these cilia. There are also ciliated cells in some multicellular organisms. For example, along the lining of the trachea and the major bronchi, you can find columnar cells with ciliated epithelium, but the flagella are mainly present in case of the prokaryotic cells, although some eukaryotic cells have flagella, like the trypanosoma, for example. All these details will be discussed later on. So up till now, it's very ambiguous. Up till now, it's incomplete data. But as we go over the cell, we are going to study more and more. As we said before, prokaryotes are mainly unicellular, whereas eukaryotes can be unicellular or multicellular, but they are mostly multicellular. We have seen now several unicellular several examples of unicellular eukaryotes. We have just said that unicellular. We have just said that eukaryotes are mostly multicellular organisms. But there is a specific group of eukaryotes, and it's important to know because you're always confused. Whenever you see a unicellular organism, you consider it a prokaryote. No, there are a few eukaryotic organisms that are unicellular, and we call them the protozoa. So what are protozoa? Whenever we mention the word protozoum or protozoa, this means a unicellular, but not any unicellular, a unicellular eukaryotic organisms. And we have several classes of protozoa. For example, we have class Sarcodina. You don't have to memorize the name, but you have to know that amoeba that is present in fresh water, it's famous in the Nile. In Egypt, for example, amoeba is, or in fresh water, this is a protozoan because it is a unicellular eukaryotic organism. And there is another class, which are the ciliata. For example, like we've just mentioned, the paramecium with the cilia all around. So the paramecium is another unicellular protozoan. We also have class Flagellata, where we have flagellated organisms. As we mentioned before, we have this trypanosoma, for example, that is a flagellated unicellular organism. And then we have the fourth class, which is the sporozoa, that can form spores like Plasmodium malaria, for example. And we have studied Plasmodium malaria before. So I'm just telling you this extra piece of information in order for you to understand clearly that it's not always about prokaryotic organisms. When the organism is unicellular, we have unicellular eukaryotic organisms. They are called protozoa, and it is important to retain the word protozoa because we are going to mention it frequently. And examples are amoeba, paramecium, trypanosoma, and Plasmodium malaria. Of course, prokaryotic cells are smaller, and as you see at the bottom of the table, that their diameter ranges between 1 and 10 microns, whereas that of eukaryotic cells is larger, and it ranges between 10 and 100 microns. And it ranges between 10 and 100 microns. Let's move on to the next slide. So here, the slide in this diagram, we show you what's common and what's uncommon between, or what's different between prokaryotic and eukaryotic organisms. Let's start by the similarities. Both of them have DNA as their genetic material, but actually, this term chromosomes is not accurate to say that it is a common thing, because we talk about chromosomes only when we talk about eukaryotes, because in order for you to say a chromosome, it should be a highly condensed means highly folded DNA material. And in order for it to be a highly condensed DNA, there should be associated with that DNA proteins called nuclear proteins. And these nuclear proteins are of two groups, or belong to two groups: histones, and we will understand later on what histones are, and non-histones. So these nuclear proteins are very important because they help tightening of the DNA and its folding and its condensation. All this is not present in prokaryotes. So we can only talk about chromosomes when we have highly condensed DNA around histones and non-histones. But we can also say that a common thing is the presence of DNA as the genetic material. Also, we have the presence of ribosomes, cytoplasm, of course, plasma membrane. All living cells have ribosomes, all living cells have cytoplasm, all living cells have plasma membrane, and sometimes a cell wall is a common structure, because prokaryotes are bacteria, and bacteria have their cell walls, and eukaryotic cells have cell walls when they are plant cells. But it's very different in structure, because as you all know, the cell wall of plant cells is made of cellulose, whereas the cell wall of bacteria is made of different structures, but mainly of peptidoglycan. Peptido refers to proteins, and glycan refers to carbohydrates. And then in eukaryotic cells, we have a true nucleus, and we have the chromosomes, because when DNA is folded, it forms a linear structure that is called DNA. So the chromosomes are linear. It is complex DNA because it is attached, as we said before, to histones and non-histones. There are organelles. There are different types of organelles, more than the ribosomes, and they are bound by membranes, whether a single membrane or a double membrane. Eukaryotic cells are typically larger. Eukaryotic cells can divide either by mitosis to increase their size and grow or heal, and meiosis when they want to form the gametes for sexual reproduction. Most of them are multicellular and most of them are aerobic. So typically, no, they are mostly multicellular and they are mostly aerobic. Here, on the other hand, we said that they can be aerobic or anaerobic. Here they are typically unicellular. Yes, I agree, because there is no single prokaryote that is multicellular. They divide not by mitosis or meiosis, but simply by elongating, multiplying their DNA, and then dividing into two. This is what we call binary fission. They are much smaller. They don't have membrane-bound organelles. Their DNA is simpler because it is not complexed to nuclear proteins. And their DNA is circular. It does not have a nucleus. It floats freely in the cell. And they have what we call the nucleoid. Oid means similar to. So the suffix oid means similar to, almost similar to, but it's not exactly the same. What is the nucleoid? We have just said that the DNA is a single circular molecule in the center, and there is a halo of cytosol that is a bit paler than the rest of the cytosol. So this DNA, together with the pale cytosol around it, is called the nucleoid, or similar to a nucleus, but it's not a true nucleus. So here you can see the prokaryotes. You can see the cell wall, and the cell wall is made of, this is the brown thing here, is the cell wall. And as I told you, it's made of peptidoglycan. Actually, they shouldn't include the membrane here. And then you have the outer membrane, and it is a cell membrane, just like any other cell membrane. And some prokaryotes, some bacteria are encapsulated. When they are encapsulated, they are more fierce, more virulent. So encapsulated bacteria are virulent because the capsule helps them be protected against the immune system. But at the same time, this capsule is very triggering to the immune system because it is made of foreign proteins and foreign molecules. Some bacteria have these pili, and we will see what a pilus is, but up till now you can say that the pilus is more or less similar to a small microtubule or a small cilium. And then we have the plasmid, and we've talked about the plasmids before. There are multiple small circular DNA molecules that are next or present in addition to the central DNA molecule, or the main large DNA molecule. They are smaller, they are multiple, they are many. They are multiplied while the main bacterium is dividing, and here we have the flagellum. Of course, not all bacteria or not all prokaryotic cells have these structures, but here we have brought you all the structures that might be associated with a prokaryotic cell or with a bacterium in order to help you understand that all these structures might be possible. In a typical eukaryotic cell, we have, of course, this is a typical eukaryotic cell because it has almost all the structures, but it's worth noting that it is an animal cell, not a plant cell, and therefore it will have no green plastids or no chloroplasts. So you can see a cell membrane, and we have said that all cells should have a cell membrane because a cell membrane controls the traffic into the cell, controls the traffic of molecules. It controls what enters the cell and what leaves the cell. Furthermore, it encloses the contents of the cell. It encloses the contents of the cell. And then we have the cytoplasm. Actually, the cytoplasm is the cytosol plus the organelles, because the jelly-like substance that exists between the organelles is called the cytosol, and it is the site of many biochemical reactions because it contains many enzymes. But when you say cytoplasm, you mean all of this. You mean the cytosol and all the organelles around the nucleus. If you add the nucleus, then it is protoplasm. So we can say something interesting here, that protoplasm means cytoplasm plus nucleus, and that cytoplasm is cytosol plus organelles. Cytosol plus organelles. And then you have all types of organelles here. So we can see, for example, a vacuole for storage of excess fluid and minerals. You can see, for example, here, let me start by the more common ones, the mitochondrion for energy production. It is the main site for energy production in the cell. You can also see the ribosomes. They are the sites of protein synthesis. We said that the cytoplasm contains enzymes and hormones, and it is the site of biochemical reactions. Of course, the nucleus controls all the metabolism of the cell and all our characteristics in general. So the nucleus controls all the metabolism in the cell or all the biochemical reactions, and therefore it controls all our characteristics through containing the DNA and the genes that determine all this. The nucleolus is a shiny small body inside the true nucleus, and it is one of the characteristics of a true nucleus. So in order for you to say a true nucleus, so you should have a nuclear membrane, and that nuclear membrane encloses a jelly-like substance called nucleoplasm, important for or site for biochemical reactions. It includes highly condensed DNA called chromosomes, and it contains the nucleolus. The nucleolus is the site it's very important because ribosomes are assembled there. They're manufactured there. So ribosomes synthesis, or ribosomes actually assembly, not synthesis. They are not synthesized there, but their components are assembled there. And then you have the lysosome, and the lysosome contains enzymes for degradation of certain old components of the cell. And then you have the smooth endoplasmic reticulum. First of all, it is important for it's like tunnels inside the cell for transport and also for lipid synthesis. But we also have a rough endoplasmic reticulum, which is also important for transport. But being rough means that it has ribosomes on the top. So it's not only like smooth tunnels like this, but on top of the tunnels, we have ribosomes, and these ribosomes synthesize proteins, and these proteins will be transferred to the Golgi body. The Golgi body will receive these proteins. It will undergo or it will carry out modification. It will modify these proteins, and then these proteins can stay inside the cell or they can be exported to the outside of the cell, and we will see examples of all this. Of course, we sometimes ask you, we give you a characteristic of a cell. For example, we give you several cells, like for example, cell A, cell B, and cell C. So for example, in cell A, we tell you that cell is rich, for example, that cell has a large sap vacuole and is rich in chloroplasts. So you deduce that this is a plant cell. Or we tell you that this cell has plenty of mitochondria, plenty of mitochondria, plenty of ribosomes, okay? So by having said that it has plenty of mitochondria, then you know that this is a very active cell. By having known that it's rich in ribosomes, so it is a cell that produces a lot of proteins. If we tell you that it is characterized by the presence of a very small cell, so this emphasizes the idea that it is a highly active cell, and we will say why. And then they will give you one or more details to tell you. For example, when you have very active cells, you think about the liver cell, you think about the brain cell, you think about the muscle cell. So if they tell you that in addition to having first of all, the fact that they are small will exclude muscle cell and will mostly exclude brain cells, and you will have the liver cell. But if they tell you that they are large cells and that they contain elongated filaments in a regular way, for example, then it is a muscle cell. Sometimes we tell you that the cell does not contain any organelles. So if we tell you that the cell does not contain any organelles, so it is mostly a dead cell, because if it were a prokaryotic cell, it would contain only ribosomes, but at least it would contain ribosomes. On this slide, we'll be discussing how different cells that have common features, as we have just mentioned. For example, prokaryotic cells have common features and eukaryotic cells have common features. But despite these common characteristics, there is some kind of differentiation between the cells, which means that some cells have a different structure, and this different structure will serve their function. And this is what we have always talked about structure, function, adaptation, which means that how the structure of the cell is adapted to carry out its function. But we sometimes ask you this question like this. For example, describe, which means explain. Describe how the structure dictates function. How structure dictates function, or how function is dictated by structure. So this is the same concept. When you have a very peculiar structure that is different from other cells, then this is because you are adapted to carry out a particular function. Inside our human bodies, there are, of course, eukaryotic cells, but these eukaryotic cells are of about 200 different types. And each of these 200 different types carries out a particular function. Why? Just because it has the structure that makes it adapted to that function. So function dictates structure and vice versa. So all cells do not look alike, and for one simple reason, they do not carry out the same function. For example, nerve cells. When we come to nerve cells, they have a cell body, and they have a long or protoplasmic projection that is called the axon. And therefore, many of the nerve cells are very long, and some of them are really long, like the nerve cells that enter in the structure of the sciatic nerve, for example, that arises from the spinal cord and supplies the whole leg with all its branches. So a nerve cell is long and spindly. A spindle-shaped cell is a rather more or less long cell, tapered at both ends, because, of course, there are, even inside the nerve cells, there are differences in structure because of difference in function. So the sensory cells will differ from the motor cells. The neuroglia cells inside the brain that form like the connective tissue of the brain and that connect nerve cells together are different. So even inside the world of nerve cells, we have different types of cells. Fat cells, for example, in general, they should be rounded and distended because they are full of fat. But even inside the group of fat cells, we have seen in human biology, for example, that we have white fat cells that consist of for example, a cell with a large fat globule inside and a crescent-shaped compressed nucleus. And it's obvious that this white fat cell has mostly the function of storage of fat, because here we are storing the fat inside that fat globule. But there are other fat cells that are brown fat cells, as we've studied before, that have a rather normal nucleus, but instead of having a big fat globule, they have small fat droplets like this, because their purpose is not storage of fat, but they are full of mitochondria because they burn these fat cells and produce energy. So, for example, brown fat cells are here in order to heat up your body whenever needed. So, but in common, they have several essential eukaryotic cells. For example, plant cells have the same essential organelles as eukaryotic animal cells do, because they are both eukaryotic cells, but plant cells are characterized by having plastids that might contain chlorophyll and be chloroplast, or might contain cyanin and be cyanoplast. They contain colored pigments, and they're also characterized by sap vacuoles. You might be asking yourself, we have just seen vacuoles in the animal cell. Yes, we've just seen vacuoles in the animal cell, but they are not considered sap vacuoles. We never call them sap vacuoles. Sap is a word that is restricted to plants, and sap here means water plus the surplus salts that are present in the plants. And the sap vacuoles are characterized by being single, large, and containing the surplus fluid and salts. And here on this beautiful slide, we can see the structure, function, adaptation of certain cells. And as we have studied before, inside your bone marrow, for example, inside under your skin, you have, and elsewhere in the body, almost in every organ of the body, but they're mostly abundant in the bone marrow and under your skin, you have stem cells. And these stem cells are characterized, as we said before, by being undifferentiated cells. And since they are undifferentiated cells, then they are unspecialized cells. But these cells are, and first, in addition to being undifferentiated and unspecialized, they are supported to be immortal cells, meaning that they can continue to divide forever unless they become specialized. So you can trigger them to become specialized. You can trigger them how? Either inside a test tube in vitro. You can trigger them through special treatment in vitro in test tubes by putting them in a special culture medium, by starving them and then giving them nutrition. There are several ways. You're not supposed to know this, but you can also trigger them in vivo to differentiate by putting them in the organ, in a special organ. For example, if you put the stem cells surrounded by liver cells in the liver, they might differentiate into liver cells. If you put them in the skin, they might differentiate into skin cells and help, for example, repair or replace burned skin, and so on. Why? Because every organ and the cells of every organ and the fluids around these cells have specific signaling molecules, specific proteins, specific hormones, specific enzymes that will affect these stem cells and help them differentiate into the same type of cells. So stem cells, when they are cultured in a special way, in a particular medium, can differentiate into several types of cells, into muscle cells, blood cells, nerve cells, cardiac cells, liver cells, and intestinal cells. So they can actually differentiate into all types of cells. And by the way, we start our life as undifferentiated cells, and then and that's why in early embryos, for example, early embryonic cells are not yet differentiated, and this is normal because they are going to differentiate later on into all these cells that will form the whole human body. So early embryonic cells are a good source of stem cells, of undifferentiated stem cells, and they are usually totipotent cells, meaning that they can differentiate into all other types of tissues because they have the burden of forming all tissues in the body. But the later you take the cell from the embryo, the more differentiated it is and the least capable to, or the more differentiated it is and the less capable to differentiate into all types of tissues it is. So we have the earliest embryonic cells are totipotent, but after having gone through several cycles of cell division, they become less capable of differentiation, meaning that they cannot give you all types of tissues as they used to do. So we have totipotent giving all, pluripotent giving many types of cells, and then we have oligopotent giving only a few types of cells, and then finally they become differentiated. And when they become differentiated, they start to form a cell line, meaning that they will give you their descendants will be the same. Here, for example, we can say examples of cells and how they are differentiated, and they are related to the question I've told you about on the previous slide, is that we can give you the characteristics of the cell and then ask you what cell it is. For example, sperm cells, they carry half the genetic material. For example, here the number of chromosomes is not 23 pairs or 46, but it is only 23. And that's why it is a haploid nucleus inside the head of the sperm, and they have tails in order to allow them to swim to the egg cell. Here, for example, we have cells that are adapted to carry oxygen. How are they adapted? By containing the red pigment hemoglobin that can form a loose bond with oxygen, and the bond is loose in order to facilitate the delivery of oxygen to the cells. It can also carry carbon dioxide back from the tissues to the heart. We also have the nerve cells, which are characterized by being very long and by being very active cells, and therefore they contain mitochondria. We also have a type of plant cells called the palisade cells, which are present in the upper and lower epidermis or outermost layer of the leaf cell. They're rich in chloroplasts because they are exposed to the sun, so they are supposed to photosynthesize, and they have to be provided with lots and lots of chloroplasts to carry out this function. On the other hand, root hair cells are also plant cells, but they do not contain chloroplasts at all. So if we ever ask you about a plant cell that does not contain chloroplast, you would talk about the root hair cell, because the root hair cell is under the soil, it's not exposed to the sun, so it's energy consuming and useless to provide these cells with chloroplast, but they are provided with long processes that help these plant root cells or root hair cells to absorb water and salts, and we call them root hair cells because these extensions that you can see on the diagram are sometimes very long and very thin. The muscle cells, as we said before, they are rich in actin, they're rich in myosin, they can change their length, contract and relax, so they are contractile cells. And then we have also talked about the ciliated columnar cells in the upper respiratory tract that have the cilia that are made up of tubules, and at the bottom of each cilium there are motor proteins that help the cilia to move back and forth like this in order to create a current and wash away mucus and the trapped organisms. So they help in immunity, in local surface immunity. The cilia help in surface immunity, which is the immunity on the surface of the mucosa, and it is a type of local immunity. Of course, you can easily differentiate here between an animal cell and plant cell, and as we said before, the three most important determinants, or four actually, we have the cell wall that is made up of cellulose. This is not present in plant cells, so this is the first difference. The second difference is the presence of chloroplasts. The third difference is the presence of a large subvacuole that's not present, and that's not present in the animal cell. And you can see that there are no centrioles in a plant cell. No centrioles in a plant cell. There is something that replaces the centriole, and we will be discussing this later on during our study of cell division. Okay, so these are the major differences between an animal cell and a plant cell. And then we're going to talk about the surface area to volume ratio. As the cell becomes smaller, the surface area of the cell, the ratio between the surface area to volume ratio, will increase. Let's look here. If, for example, the side of the cell here is 3 units, 3 microns, for example, then the surface area of each side, suppose that the cell is a cube or cubical in shape, so it has 6 faces. The surface area of each face will be side by side. It will be 3 by 3, meaning 9 squared centimeters. So if you multiply 9, which is the surface area of one face, by 6 faces, then you obtain 54. But the volume is not side by side. The volume is side by side by side, meaning 3 by 3 by 3, which is 27. If you divide the surface area by the volume, you obtain, if you divide the area, which is 54, by the volume, which is 27, you obtain the surface area to volume ratio, which is 2. But if the side is 2, then 2 by 2 make 4. This is the surface area of one face. 4 by 6 faces gives rise to 24. So this is the total area, whereas the volume is 2 by 2 by 2, side by side by side, which is 8. 2 by 2 by 2 equals 8. If you divide 24 by 8, you get 3. So 2 is 54 divided by 27. 3 is 24 divided by 8. And if it is only 1, the ratio will be 6 divided by 1. So the ratio of the surface area to volume increases as the side of the cell decreases, or as the size of the cell decreases. So if you have a smaller cell, then this cell will have a higher surface area to volume ratio, but it will have a smaller volume to area ratio, because we sometimes give you this. What's the importance of this? What's the importance of the smallest cell having the largest surface area to volume ratio? This is important because it is through the surface area that there is transport of material inside and outside the cell. And that's why active cells should be small, because when they are small, we don't say that never say that they have the largest surface area. Actually, they do not have the largest surface area, but if the cell is small, it will have the largest ratio. of area to volume. So it's totally incorrect to say that smaller cells have largest areas. No, because the surface area of the smallest cell here is 6, but they have the largest surface area to volume ratio, and this allows them to transport material in and out. And it's needless to say that active cells require to get substances in for their biochemical reactions and to get substances out, whether these substances out are side products, waste products, or useful products to be transported to other cells. So when the cell transports substances in, so transport can be in or out. Why do active cells require inward transport? In order to get the ingredients, in order to get the raw material or the substrates that are needed for the chemical reaction. But out, in order to either deliver the products, the useful products that they have formed, or to get rid of the wasteful products. So this is why active cells should have a surface area to volume ratio good for transport. You might be asking yourself, why isn't the active cell just having a large surface area and that's it? Why it is important to consider the surface area to volume ratio? Because if you want your cell to maximize the transport of material just by having a large surface area, it would be a very huge cell. But you want that cell to increase its power of transport in and out, but at the same time minimizing the size so that you can have a greater number of cells in the organ and therefore work out more efficiently. So this way you are increasing the efficiency of the cell. So by increasing the surface area to volume ratio, you are increasing the power of transport in and out, or actually you can say the power of exchange, but meanwhile without occupying a huge space. We want this active cell to be small in order to have more of it in the same size of the organ and therefore increase the efficiency of the organ. So for the surface area to volume ratio, the surface area of the cell membrane of the living cell must be able to adapt to the metabolic needs or to accommodate the metabolic needs based on the volume of the cytoplasm. That's why multicellular organisms consist of tiny small cells having specialized functions. And how does the cell divide? You will be amazed when you find that this surface area to volume ratio determines the timing of cell division. This is an important concept to understand. The surface area to volume ratio, determines the time of cell division, because when the cell takes in substances and gives out substances, this cell will grow in size and the volume of the cytoplasm will become too great for the area of the cell membrane, meaning that when the cell receives nutrition, when it receives glucose, for example, and gives rise to energy, and the cell carries out chemical reaction, the daughter cell at the beginning is not large in size. This daughter cell requires to grow in size. So it keeps growing in size until the surface area to volume ratio is no more good for the metabolic rate of the cell. At this time, it has to divide again in order to have a higher surface area to volume ratio and allows more transport. Okay, so here we have a daughter cell. and then that daughter cell receives nutrition and gets rid of waste, for example, or for sure, and then it grows in size. As it grows in size, what happens? The volume increases. So what happens to the surface area to volume ratio? It would decrease. And at that time, there is a major decision to be taken. We cannot continue like this because as we grow in size, our volume increases, and our volume increases, so the ratio decreases, and we are no more capable of receiving adequate transport. Okay, so for example, active cells like the liver and brain are smaller, and if they are big, they would be long but not thick. They'll be just elongated, as if you have a spherical cell and you keep the same volume but you stretch it out to increase the surface area the surface area per unit volume, and therefore make it more efficient in exchange and receiving nutrients and getting rid of wastes. In this slide, we are going to differentiate between organelles with no membrane at all, organelles with a single membrane, and organelles with a double membrane. And here, of course, in this table, we are assuming that the nucleus is one of the organelles, the largest and the most important. So the nucleus, the mitochondria, and the chloroplast have a double membrane. So these are 3 organelles that have a double membrane. But we have 5 organelles that are not membraned. The ribosomes, the nucleolus that is present inside the true nucleus, the myofibrils, which are protein structures inside myosin and inside the muscle fibers. So a myofibril is the smaller structure or the substructure of a protein fiber in general. For example, a muscle cell or a muscle fiber. And then we have the cilia. These cilia, we have seen before, are not membraned, and the centrioles. What are the centrioles? They are seen in animal cells next to the nucleus. You can see 2 cylindrical bodies that are perpendicular to each other and that are very important in cell division, as we will see later on. All the rest, endoplasmic reticulum, Golgi, vacuoles, lysosomes, peroxisomes in plant cells, vesicles, acrosomes in sperms, melanosomes in skin cells. So acrosomes are actually present in sperms. Melanosomes are present in melanocytes of the skin. Peroxisomes are present mainly in plant cells. All these have a single membrane. So it's easier for you to study you already know the 3 organelles with a double membrane. They are very famous. You only have to study the 5 organelles that do not have membranes, to start by a C, centrioles and cilia, M and N, myofibril and nucleolus, and then finally, of course, you all know that ribosomes do not have nucleus. By having known these 8 organelles, then the rest of the 16 organelles are all having a single membrane. Now we are going to talk a bit about the nucleus. Of course, as we said before, the nucleus is, in my opinion, because in my opinion, the nucleus is one of the organelles of the cell. The nucleus has a double membrane, as we said before, and the nuclear membrane or the nuclear envelope has pores. And these pores are very important because they will allow, the simpler thing to tell you is that they will allow messenger RNA, which is the representative of DNA, to leave the nucleus and go to the cytosol to be translated into a peptide, because translation occurs in the cytosol. But making RNA on top of DNA, which is transcription, occurs in the nucleus because DNA cannot leave the nucleus. So messenger RNA is one of the substances that leave the nuclear pores. Also, the nuclear pores will help ribosomes to reach the cytosol, because ribosomes are the sites where messenger RNA will be translated, and ribosomes are collected or gathered or assembled in the nucleus. So there are two things that, or two important structures that leave the nucleus through the nuclear pores, and these are an organelle, which is ribosome, and one type of nucleic acid, which is RNA or ribonucleic acid. And as we said before, the nucleolus is a shiny, non-nucleated body inside the true nucleus, and it is the site of assembly of the ribosomes. Why assembly of the ribosomes? Because inside the nucleus, transcription does not only give messenger RNA, but transcription also gives rise to another nucleic acid, which is ribosomal RNA. And it is called ribosomal because it is an essential component of this organelle, which looks like a burger bun, and that is called the ribosome, made up of a large 60S subunit and a small 40S subunit. But ribosomal RNA is an essential component of the ribosomes, and it is made inside the nucleus via transcription. But meanwhile, we still need polypeptides to be assembled with this ribosomal RNA in order to make a ribosome, a complete ribosome, and before the ribosome leaves the nucleus. These polypeptides are, just like any peptides, are synthesized in the cytosol, and we call them ribosomal polypeptides. So just like the nuclear pores give or allow the fully made ribosomes and the messenger RNA to leave the nucleus, they also allow the ribosomal polypeptides that has been synthesized outside the nucleus in the cytosol inside other ribosomes to go back to the nucleus and be assembled with ribosomal RNA to form a ribosome. So it's strange that the ribosomal polypeptides, it's a bit weird. They're made inside ribosomes, already made ribosomes, and they return after having been released from the cytosol ribosomes. They return through the nuclear pores to the inside of the nucleus to be assembled with the ribosomal RNA that resulted from transcription on top of DNA inside the nucleus. Once they are assembled, they form freshly made ribosomes that will leave again through the nuclear pore. So the nuclear pore allows the polypeptides in, and then it allows the ribosomes out. The nucleus also contains, of course, DNA, which carries the code for genetic information, and this DNA, as we said before, is highly folded or wrapped around nuclear proteins that are called histones and non-histones to form the chromosomes. But the chromosome is the final fully condensed form of the DNA. But once DNA is wrapped around histones and non-histones, we obtain chromatin. Chromatin is nothing but DNA plus the nuclear proteins, histones and non-histones, in the ratio 50-50 or almost 1-to-1. And then this chromatin, so whenever we say chromatin, we mean DNA plus nuclear proteins in the ratio 1-to-1. But this chromatin needs further condensation and folding to form chromosomes. The DNA in these eukaryotic nuclei is surrounded, as we said, by the nuclear membrane, because it's a true nucleus. And we can say that the nuclear envelope or the nuclear membrane is selectively permeable. Why? Because it contains, as we've explained, nuclear pores, and these nuclear pores allow certain molecules to pass, like the ribosomal polypeptides, like the fully formed ribosomes, the newly formed ribosomes and messenger RNA, but it can prevent other molecules that are too large or that are harmful to visit the nucleus. Okay, inside the nucleus, as we said before, we have a shiny body which is called the nucleolus, and that shiny small body is not surrounded by a membrane at all, and it is the site where assembly of ribosomal RNA and polypeptides occurs to form fresh ribosomes. In some nuclei, when the cell is very active, like in certain liver cells, you might find 2 nuclei, or you might find 1 nucleus with 2 nucleoli. The nucleoli, as we said, contain chromatin, ribosomal RNA, and unfinished ribosome precursors. So the nucleolus, as we said before, consists of ribosomal RNA simply and ribosomal polypeptides. So actually, since it is nucleic acid, even if it is RNA with a polypeptide, then it is chromatin. That's why we say that the nucleolus consists of chromatin. But you know that the chromatin inside the nucleolus is different from the chromatin outside the nucleolus. The chromatin outside the nucleolus is what? The chromatin outside the nucleolus is DNA plus histones and non-histones. But this is outside the nucleus. But inside the nucleus, the chromatin consists of ribosomal RNA plus ribosomal polypeptides. And this chromatin is considered an unfinished ribosome precursor because it will undergo several modifications to reach the final stage of the finished ribosome. As we said before, ribosomes are composed of 2 subunits like this, and these 2 subunits are sometimes called the 50S subunit and 30S subunit, or they might be called 60S subunits and 40S subunits. And the S here, actually, we say it's subunit, but it refers to a unit called Svedberg unit, and Svedberg unit is a unit of density. So the 50S or 60S subunit have higher density than the 30S or 40S subunits. The 30S subunit, for example, consists of 16 ribosomal RNAs and 21 proteins, whereas the 50S subunit contains 5S and 23S ribosomal RNA and 31 proteins. This is not at all important. This is very complex and you needn't know all this. You just have to know that the larger subunit has a higher density, that the lower subunit has a lower density. And then we are going to talk about peroxisomes. Peroxisomes are organelles that are abundant in plant cells, but that are present in both animal and plant cells. And the peroxisomes are characterized by having an enzyme, very characteristic enzyme, that is called the catalase enzyme, that catalyzes the decomposition of this molecule, which is H2O2 or hydrogen peroxide, into water and oxygen, meaning that it will convert a molecule that is mostly harmful into 2 molecules, one of them, or actually 2 molecules that are useful. Why is H2O2 or hydrogen peroxide? Here it is called peroxide because it is water that has an extra oxygen. Why is it produced? It is produced as a byproduct of respiration. So you cannot prevent or help the formation of peroxisomes. Not only do peroxisomes convert this harmful or mostly harmful H2O2 into water and oxygen, but they also help detoxification of alcohol, conversion of alcohol, which is a toxic substance inside liver cells, into a non-toxic substance. And that's why you are frequently asked, when we tell you that a cell is small, very active, and it is an animal cell and it is rich in peroxisomes, so it might be the liver cell. A third important function of the peroxisomes that have, as you see, one lipid bilayer membrane, as you see in the upper diagram, it is the breakdown of very long chain fatty acids into medium chain fatty acids. Why do we need to break down the very long chain fatty acids into medium chain fatty acids? Because it's easier for the smaller medium chain fatty acids to move to the mitochondrion, to be shuttled to the mitochondrion, to enter the mitochondrion, and be broken down to get water and energy, and be broken down in order to get water and energy. Okay, so we will stop here, and we will continue our structure of the cell in another video. Thank you and goodbye.